So once again, good morning, everyone. As I have mentioned that we have with us Dr. Ranjeet Kumar. He is PhD from CDRI Lucknow and currently working as a senior research engineer at Chalmers University of Technology, Gothenburg, Sweden. And his research is focused on understanding the molecular events involving protein folding, misfolding, and disease manifestation. So, sir, I'm not going to take much of your time,
Yeah, okay.
and he'll be, explain you about his journey and all the details of the given topic.
Mm,
So, on behalf of Srishti and Honeybee Network, I welcome you, sir. Over
mm.
to you.
Thank you, Megha, and thank you for inviting me again to deliver a talk in this webinar series meant for Biotech Innovation Ignition School. Actually, I was here in December, if I remember correctly, for a lecture, and I'm using the
Yes.
same lecture more or less. So,
Yes.
so the lecture is titled "Protein Factories: Expression, Purification, and Application." So before I take you to the lecture, I would like to give you a brief introduction about myself and my journey into the field of life science. Hope I am audible.
Yes.
Okay. So so I will take you to through my Milky Way of knowledge and how I uh, traverse the journey from becoming to being a scientist. So initially, I would owe everything to my mother and father, who are like the center of the galaxy for me. My initial schooling has been very since my father was into administrative service and with frequent transfer, my actual schooling started in grade 6 or class 6, where I joined St. Xavier's School. Uh, and after that, St. Xavier's School in a small town called Hazaribagh. I belong from Hazaribagh, Jharkhand. And then I studied. It's a small, beautiful town uh, coming in Chota Nagpur Plateau. And the best thing about this city is that there are many good colleges and institutions. And that's why my father decided to settle there. And that's how we got to get very good education at that time when we were like hopping from one city to another because of his job responsibilities. So here you see my father and my mother. And then I ended up going into a college which is called St. Columbus College, and they were starting a program very new at that time. It is like, I think it was 1998 or so, a program on biotechnology. And at that time, biotechnology was a lesser heard word. There were not many institutions pioneering this, and I think St. Columbus College was one of the 12 places chosen by the government of India to start a bachelor program at that time in biotechnology. So eventually I entered into studying biotechnology. And a good thing about this program was that uh, we got to get very good on the job training and summer projects. And this was our first introduction to the field of biotechnology. Uh, and one good thing that happened is that I also uh, uh, got to get several trainings in this period. Then it was a part of a university now known as the Vinoba Bhave University. And then after the bachelor's, there was a lot of turmoil. Like we didn't know where to go for the master's because there was no structured information available for the course. And the only way to move ahead was to, like when I did my bachelor's, there was kind of, okay, we did study something, but it doesn't lead to anything at that present moment because there were very few industries. And being from a small town, there was also lack of information and we didn't know what exactly we need to do now. So the only option available was to sit for an exam which was conducted by Department of Biotechnology and JNU combined. And if you qualify that examination, then there were like 30 or so, I think 24 or so universities which were offering master's program in biotechnology. And that's how I ended from like being uh, studying in north to going to the extreme south of the country. I went to Calicut University for my master's, and in the Calicut University, the program was very well designed, and the experience that we had from our bachelor's helps us a lot. And there I got to get training in Institute of Life Sciences Bhubaneswar as a part of my project work, and also in summer training I visited Institute of Microbial Technology Chandigarh, and this was like an eye-opening and setting up of the stage that what's going to unfold in future. Then as every like postgraduate would do is that again at the postgraduate stage we were feeling that there is no opening at the master's level to go for, like the industries are few, it's not well paid. So what's the next step eventually? So it was like, let's crack this exam which is called either graduate aptitude test for engineering or uh, junior research fellowship, which is conducted by Council for Scientific and Industrial Research. And then when we cleared this exam, I for a short while, because I I got a bad hepatitis at that time and couldn't appear for many of the like interviews. So I ended up joining a ICMR institution known as National Institute for Research in Reproductive Health in Mumbai. Worked there for almost 6 months, and then I reverted back to some place near my house because it was year 2004 and my mother expired, and then there was like a lot of turmoil and personal problems. So I reverted back from Mumbai to Central Drug Research Institute, Lucknow, and I started my PhD under the mentorship of Dr. Vinod Bhakuni, who who was working in the area of protein folding, misfolding, and understanding proteins. Um, Dr. Bhakni is no more, but I owe a significant part of my personality to him. He was a Santi Swarup Bhatnagar awardee. And then after my PhD from CDRI, in the due course of PhD, it was a very exciting time. We got kind of good exposure and training, and I also got to participate in some program on entrepreneurship. Especially, there was one very structured program known as Technology-led Entrepreneurship Development Program run by CSIR. And this was an eye opening because it was more towards how do you connect science with the society. Finally, I got my degree from JNU, the PhD degree, and the first thing I did was that there were many who were opting to go to US, but somehow my boss had a feeling that every student of science would have a working knowledge of Europe, because in Europe there is the situation is different because there is a lot of uh, they believe in collective excellence. So I ended up coming to Uppsala University, which is one of the oldest university in Scandinavia, and started my work. After a year or so, I realized that I am not that fit, and there was again coming from India, I had some adjustment issues regarding the weather, and the project was not turning the way I wanted it to turn. So I moved to a small university again in Sweden and changed my project to environmental biotechnology, where we were uh, uh, like from a protein science to hardcore molecular biology, developing tools and techniques to study environmental degradation. And then I went to Russia. Actually, I went to Siberia in a place called Tomsk, and with a scientist who was authority in copper as antibacterial, and I worked with him on a project called nanopumps. And then I developed some interest in Ayurveda, and the reason being the link between copper and Ayurveda. So I studied a small diploma to understand what goes on in the in Ayurvedic system. And then I reverted back to the same university from where I did my bachelor's actually. And I got a faculty position. I started there as a assistant professor. But soon I realized, and I was teaching both in the college and in university out there, I realized that the research infrastructure which I am used to over a decade is not that well established in the state universities. And my hunch for doing good research was still there. I wanted to do research as well as teaching. And to be very frank, I don't like to sit and give lecture. I want to work in the lab. So that brought me back to Chalmers University, where I am working right now. I work as a senior research engineer, and this gave me again an opportunity to revert back and work on the area where I wanted to work. So, and when working in Sweden gives a very good work-life balance, and I get a good opportunity to train myself in skills at various university, as well as contribute largely to global science scenario. Uh, so here I am, Dr. Ranjeet Kumar, with a MSc Biotechnology from Calicut University, Calra, and PhD from CDRI Lucknow, and currently working as a senior research engineer with a research focus basically on folding and misfolding and disease manifestation resulting from malfunctions of the protein. And we here are interested in studying small molecules, proteins, and other intervention that can be made to dampen the process of protein aggregation, which is a hallmark of both the Parkinson and Alzheimer's disease. With this short introduction about an amyloidogenesis, that's the formation of plaques in the brain and formation of amyloid. With this introduction, I would like to now start my lecture. And I feel that this introduction is important because no matter from where you come or what you do, it's the constant feeling of upgrading yourself and getting to know the new things that's happening in your field, which will propel you uh, to explore uncharted territory. So now when we talk about and when we come to the topic, when you look inside a cell, and this is a representation from a bacterial cell, what you would see is that the bacterial cell is like overly crowded with different kind of macromolecules sitting within. And this phenomena is called macromolecular crowding. Because you have RNA, proteins, other molecule, protein synthesis is going on, and there is a lot of symphony of many metabolic processes that that occurs together within the cell. So what we as a chemist want to do? So it's a kind of a oversimplification that within the cell there are carbohydrates, other molecule, nucleic acid, ribosome, and you have your taste protein. There are also many other protein. But to study these protein in a in vitro environment, what you need is to isolate this protein and work on it. So here comes the whole exercise of protein purification, actually. So going a little bit into the basics. So it's like, how is the primary structure of the protein? The primary structure can be equated to alphabets. The secondary structure, which is alpha helical and beta sheet structure, can be equated to small words. Tertiary structure can be equated to sentences, and quaternary structures are like paragraph. So a protein has four different level of organization. The primary structure is just the chain of the amino acid, but due to different interactions that undergoes within to give it a stability, it can form either alpha helix or pleated sheet or beta pleated sheet. And along with it, it can also, there can be some unstructured part, which is called random coil. And then the protein assembled together. to form a functional unit or the tertiary structure, where the beta pleated and alpha helical structure arrange into a specific shape and form. And some of the protein are functional at this stage only, but there are proteins which which can function in association with other protein either by forming a hetero or homodimers or trimers or tetramers, that is oligomers. Like hemoglobin has 4 unit of globin with a heme moiety sitting in between. So the final structure. So like this is all about, like there are two kind of protein. One is this protein which is globular in shape, and there is also other protein which cannot be globular, which is fibrillar, like collagen and um, actin and myosin. But there is also some protein which are known as intrinsically disordered protein. And intrinsically disordered protein are the one which have pathogenic consequences. And what you see here is a atomic force microscopy where you can scan your sample and see the surface morphology of the protein that is present. And here what you see like a rope like structure in the golden color is the protein alpha synuclein, which aggregate in the form of a rope like structure or a thread like structure like a spaghetti. So these proteins, when they interact with membrane or lipid, they can gain some structure. Here you see the alpha helix, but largely it's random coil. And then they arrange in a specific manner to form like a, antiparallel beta pleated sheet structure, which forms the backbone of the fiber. So the to sum up that there are, you can call it three different genera of protein present in the body. One is the globular protein, like you have many enzymes which are globular, and they have a definite shape and a proper hydrodynamic radii. And then you have proteins which are like fibrillar, like actin, myosin, collagen. And then you have a group called intrinsically disordered protein, which doesn't have any shape or size of their own, and they can be a little bit notorious in long run, especially what we see in case of neurodegenerative disorders. So the question is, why do we want to purify protein? One thing is that we want to purify protein because if we want to study the process of catalysis and understand what happens both in cells and organism, we need to have a purified protein in hand. Second is that we, if we want to study the structural elements that gives the backbone to the cell, or that gives the uh, that gives the structure uh, and stability to the cell. We would like to study those elements, how they form, how they network. Then we want to study the receptors, because these receptors, they interact with ligand, and sometimes this receptor ligand interaction, if it's broken, then, or if it's not working properly, it can manifest into several form of malfunctioning, and it can lead to diseases. We also want to understand how does the intracellular signaling cascade work. Supposedly, if there is a external environmental trigger, what happens downstream? Like say, oh, in the case of allergy, if you have hay fever or some other ailment, what happens? Some kind of external agent or a pollen can trigger this kind of reaction, and there is a intracellular cascade and signaling, which ultimately um ultimately uh, instigates or starts the uh, immunological response, thereby leading to redness of eyes or uh, uh, um, slight fever and those kind of manifestations. Then one also, there are several key components which are proteins in different machinery. If you take protein translation, there are ribosomes, there are tRNA, mRNA, and there is a whole battery of enzymes and protein which facilitates this process. So it becomes important to study the protein. Also, when like if from 2 or 3 decades, if we see there has been a lot of advancement, be it in the CRISPR-Cas or be it in recombinant DNA technology, that one can manipulate DNA and RNA to an extent with the help of enzyme that you can have any desired protein or product of your choice. You can silence gene and do uh, many biochemical, you can block some biochemical circuits. So this is possible because of enzymes and protein. So I'm just presenting you a old data, but this is how the value of gold, if you see in 2011 in US dollar, was 48 per gram. But if you see the value of protein, say growth hormone or granulocyte colonies, stimulating factor or insulin, per milligram or per gram of the protein still cost higher. To sum up, uh, there is a great value for the purified protein, and I will tell you at the end of the lecture that there are many spin-off and companies which work in this area. So to sum up, it's like the biological activity because of which you need to study the protein. As a biochemical reagents, they are important in the lab. If you want to study the enzymology, the kinetics, how the enzyme work, or if you are looking for a drug for some enzymes which is malfunctioning, you need this. You need also protein purification for studying structure function studies and protein engineering, and especially drug designing when it comes to like designing new drugs for many of the diseases that are very relevant to us. So once you understand the protein or the cause behind, we want to develop inhibitors for that, and then you need bulk protein to study it in the mass spectroscopy, NMR, or crystal structure. And I think there will be several lectures in this regard, and you will get to know more. So when you want to express protein, one can choose different kind of system that's available. The very like kind of a naive system that's present in most of the lab, uh, pretty standard, is E. coli. But then you can have requirement to work with yeast, baculovirus, mammalian cell, and I have missed one thing here: plant cells. The protein can be expressed in different format. But having said that, there are certain limitations and bottlenecks of each system, and then you choose your system depending upon what is the end product and what is your need. For example, in E. coli, you can express any of the protein. You can get protein very fastly when you express in the E. coli. You can get bulk quantity of protein. But the bottleneck here is that you don't get any post-translational modification. So that that is needed, especially if the protein is of eukaryotic origin. Then you have to resort to either yeast, baculovirus, and mammalian cells. And when you do, like the best yield that you get is from yeast, from E. coli, but you can get certainly good amount of protein from yeast also. And in yeast, you cannot have all the modifications. You cannot make therapeutic protein in yeast, or you will have to do kind of protein engineering and also a little bit of uh, mm, engineering the cells in certain way to make your protein so that it's good for therapeutic use. But for food and feed, certainly you can use yeast to express protein. And also there are like many spin-off where algal proteins are being used as supplements, and I will talk about that later. Then you have a baculovirus system, which is a good system to express eukaryotic protein, and you can secrete the protein to the medium. You can have low to high concentration of the protein in this system. It facilitate proper folding of the protein, but then also you cannot have all the modification like gamma carboxylation is not possible, but most of the modifications you can have in all these three systems. So essentially, when we talk about vaccines or when we talk about proteins that are good for therapeutics, it's mostly done in mammalian cell or eukaryotic cells, especially mammalian cells, where the protein is expressed, and uh, good quantity protein uh, is not only the quantity but the quality also matter. Because if you express the same protein, say in E. coli, what will happen? That first thing is that you will not get desired modification, so the body will treat it as foreign. The other bottleneck is that E. coli has several endotoxins associated with it, so it will pass through the you can remove it by certain means, but there is always a danger of carryover. And the last, which is not mentioned here, is the plant cells. It's also a very good expression system, especially, and very well worked out because of the model plants that we have, like Arabidopsis, Thaliana, and tobacco, where it's well deciphered how they work and the genetics is known. So I have a colleague who works uh, in like there is a large section, especially the developing in the developing world, people are anemic, and he has thought of a strategy to express hemoglobin in plant. So if you express hemoglobin in plant, you get the same protein. You don't need to eat any meat or meat product, but you will get significant amount of uh, iron, and it will be good for the anemic patient. But it's still in trial, and he has been partially successful uh, in uh, in that approach. So basically, if you go with most of this current strategy to express protein are based on recombinant DNA technology, and the core of this technology lies in the basic that we studied in class 10th and 12th about the lactose operon, whereby there is a repressor which sits at the operator site. And once it is sitting at operator side, it doesn't allow any transcription to happen, as a result of which the lac Z, Y, and A, which is like sitting on the same cistronic system, because we don't have introns in bacteria. So genes are aligned one after other. So these three enzymes, beta-galactosidase, permease, and transacetylase, are not produced here. But once you have an inducer in the form of lactose, or uh, in the form of a gratuitous inducer in the lab, which is called IPTG, isopropyl thiogalactosidase, it can bind to the repressor tetramer, and it doesn't allow it to bind to the operator side, as a result of which the process can trigger the process of transcription and eventually translation, and result in the production of the three proteins that are there. So what essentially is done is that you just chop off the flag ZYA area, and you can put your gene of interest, which is under control of an inducer. So once you induce the system, the total cellular machinery of the cell will, like, apart from minimalistic survival, all the energy of the cells will be directed towards making your protein. And that's how any overexpression system works, be it a prokaryotic or eukaryotic. The basic fundamental thing is this. So if we go for a schematic, it's something like this: that first you have to clone the gene of your interest into a uh, into a plasmid, which is called vector. And in this vector you need to put inside the bacteria, which by a process called transformation. And chemical transformation is where you treat these bacterial cells with a very low concentration of chilled calcium chloride, and then you put your plasmid, give a heat shock for 30 to 45 seconds, and then you snap chill the cells, and the plasmid is taken by the cell. And then you grow the culture, induce the culture, pallet the culture, and then you undergo several steps of chromatography, which forms second part of the lecture. And then you have your purified protein in hand. So this is one part of the thing. So basically, what do we do in the lab? So expanding this schematic, you take the bacterial cell and you grow a large culture, induce it, then you harvest the bacterial cell. Then you need to break the pellet it, take the pellet, put it in a specific buffer, and then you do cell lysis. And we will see each step separately, uh, what is happening. And you can break open the cell. After breaking open the cell, if the protein is a soluble protein, it will end up in the soup, which you can load onto the column. And the process nowadays is largely automated, and I think it's pretty much same whether you work in a lab setup in India or abroad, because these things are more or less standardized globally. So then you have a automated system whereby you can load your protein and you get your purified protein, which can be checked through mass that whether you are getting the protein of your interest or not. So we also have two system here, and I would like to tell you one thing is that this system, which is from G, now the company is called Sitvia. This system is the exactly same system I have been using a decade ago in India in my lab in CDRI Lucknow. So, to why do I say so is that the lab and the infrastructure in India is at par with the international setup. So, when you transition like across the globe to any of the lab, pretty much it's more or less standard. So we have two system. One is from GE and the other one is from Biorad, and these are like automated system whereby you can load your protein, hook your column, and then you can automate the process and finally get your protein in hand very quickly. So how do these processes start, or supposedly if you have a gene of interest in your mind, how to go about it? So maybe this slide is not very clear, but you can make a note that there is a open source program called ProtParam XPASI. It's a tool whereby you can put the sequence information of your protein that you want to work with, and then it will give you all the parameters that's important for that protein. And I call sequence as the information black box of protein. So once you have this sequence in hand, if you put this sequence here in the query box and compute the parameter, it will give you all the details. And these details are mainly like uh, about the different amino acids that are present in the protein, how many of them are there, is there any tryptophan or not, what is the pi of the protein, what is the exact molecular weight of the protein, what is the extinction coefficient through which you calculate the amount of the protein, all these, and what is the stability index of the protein, every single information you will have. And I tell you that before you start any purification strategy or process, this information is very, very crucial. So you get a really good insight onto your protein, whether you are working it for a first time or if you have some known protein in hand on which you are working. This information really gives you a great insight of on how the protein will behave and help you design your purification strategy. So let's see different steps and how we do this process. So first is to extract the protein, and for the extraction of the protein, one thing is that if the protein is extracellular, say the cell is secreting the protein out into the medium in which you are growing the cell, then the process is fairly simple. You just collect the soup, remove the pellet, and you concentrate the soup in various means to finally get your protein. The other issue is that if the protein is intracellular, what you will do? Be it animal, plant, or microbe, you will have to disrupt the cell to get the cytoplasmic content of the cell out. And you can do this disruption by a galaxy of available means that is there, either by chemical method like detergent, alkali enzyme method, lysozyme method, where you can cause punch and holes in the cell, and then the content of the cell will ooze out, which you can collect. But the most common method is the physical method through which you do the thing. And here you see two systems. One is here you break open the cell through sonication, that is the sound wave. And here you break the cell. This is called French press. And you break the cell under pressure. Like you put a lot of pressure on the cell and the cell ruptures. So in both the cases, both the physical means, what we do here exactly in the lab is combine the chemical as well as the physical means to get a good yield. And the most common thing that is used within the lab, if it's not a membrane protein, we go with the sonicator approach because it's fairly easy. You just take your sample and you take it into a ice jacket, like you put some uh, ice around the system so that the sonication, when the sound waves are triggered within the solution where your cells are sitting, then there is compression and rarefactions of the waves, and because of this uh, continuous compression and rarefactions of the wave, the cells will break open and it will release its content within the medium. So these are the way you want to break the cell. And an important thing is that whatever you do in the protein purification process, your idea is to do it in such a way that the environment which is within the same cell is maintained, and the protein that you are working with is not chopped up as a consequence of the method that you are choosing to purify the protein. So you need to have certain additives and components. The most important thing is the buffer. So if you study your protein, and if you also uh, mm, do this study, like the prot param, you will get to know which buffer would be the best suited for your protein. In most of the condition, tris is the standard buffer that we use, sometimes phosphate also, to purify the protein as an initial buffer. So um, a buffer is a kind of a solution which can resist small changes in the pH, and thus will help your protein sit in the right kind of environment. Uh, and there are certain factors and some pros and cons of using each buffer. We stick mainly to tris buffer in our lab, or if there are issues, we go to phosphate. So uh, so when you are using buffer, essentially what you want to do is to have the similar kind of environment that's present within the cell and where the protein is sitting. But once you break open the cells, then there is a change in environment, because essentially everything within the cell is under a reduced environment. You don't have oxygen, like free oxygen undergoing the cells, and that's why you would have heard that you need to take antioxidant. Why do we need to take antioxidant? We need to take antioxidant to we know that if there is oxidation within the cell, then some protein will be malfunctional. That's why there is a lot of like they will say that uh, eat a lot of carrot, means in a bulk term. I am telling it, it's not at the cellular level, but at the cellular level also same thing happen. If you give a oxidizing environment to the cell, then most of the protein will stop functioning and they will malfunction. So the main additive is that you want, when the protein comes out, you still want to have a reducing environment. For that you either use DTT or TSAPE or BME. And my friends, BME is something which we avoid because it has a very pungent smell, and if anyone opens the bottle in the lab, everyone has to suffer actually. So what we use basically is TSAPE or DTT. Even if you open it within the hood, it smells a lot. And this protect the protein from oxidative damage. Then you need to, when the protein comes out, there are many proteases which are there in the system which will, like, non-specifically chop your protein. So you need to shut these proteases off, and to help in this, you have to add a protease inhibitor cocktail we call. So it's a cocktail of uh, leucopeptine, pepestine, and other commercial proteases, which we add one tablet each. You can either, like the simple way is to add a PMSS F, because most of the proteases are serine proteases, and which can be inhibited by uh, a compound called PMSF. So you need to add a little bit of it. But what we do here is we just add a tablet. Then you need to have metal chelators, because these are also nuisance in the system, because they facilitate many of the metalloenzymes or metalloproteases. And if they are there in the system, what will happen that the metalloproteases will be very, very active. And within, like in the due process of purification of the protein, you will see that your protein is degraded. And when you run it on the gel, you will not be happy to see that you don't have any protein at the end, although you followed all the procedure that was needed. So it's very, very important. Then sometime you need to have a osmolyte to stabilize protein, and the most important osmolyte that's used is glycerol. But there are other osmolyte, like small amount of detergent is used, especially when you work with proteins that are associated with vesicles, or those which are associated with membrane. So to stabilize those protein, you need that kind of a system. Then you also need ionic stabilizers, and ionic stabilizers are very, very important. Especially we, like a blanket case, we add 150 millimolar of sodium chloride into the system to enhance the solubility of the protein. But sometime we have seen through experience that some of the salts can be either cosmotropic or chaotropic. Chaotropic is like something which generate chaos in the system. So if you can use those salt when the protein is not soluble, or it goes into the pellet, or it forms some kind of a inclusion bodies within the cell. So to extract those protein, you have to use chaotropic salt. But salts like sodium chloride or potassium chloride and ammonium sulfate, they increase the solubility of the protein. And one way to do is, yeah, and which is a pretty standard and an easy way, whether you do recombinant DNA technology, a protein production through heterologous means, or if you are purifying protein from a natural source which is rich in that protein, this ammonium sulfate precipitation is a very good step. So what happens essentially is that your protein is hydrated in water. So once you start adding ammonium sulfate salt and increase the concentration from 0 to 100%, what will happen that this salt will associate and form hydroxide and will exclude the water. As a result of this, the protein molecule will get together, uh, and then they will clump together due to the interaction, because the water is excluded, and they will ultimately, you will precipitate the protein. And you can precipitate, and this process is called salting out. The same phenomena happens because one easy way to do water purification is to add uh, we call it, I don't remember the English name, but it's called fitkiri. So you have this salt that you add, and you just put a little bit of it, and if there are a lot of dirt and things in the water, it will settle down, because this the same phenomena happens. So the salt just removes all the debris that's sitting in the water. So this is also a very important step. I will stop here for a while, and if you have any question till now, you can ask me, and then we go to the second part of the lecture where I introduce you to column chromatography. Yes.
Excuse me, sir.
Yeah.
Sir, you mentioned about like hemoglobin expression of hemoglobin in plants.
Yes.
Yes, sir. It didn't find any resistance. So, like cells and non-self cells, such type of resistance was not found
I didn't
in
get your
plants.
question.
So,
Uh,
like it was expressing hemoglobin in plant cells, huh?
yes. Yes. Yes.
So
So
hemoglobin. Yes, sir.
So the way you do it is that you use recombinant DNA technology. So you you make certain viruses which can infect plant cells, and these viruses, they are designed in such a way that they have your gene of interest, which is the 4 unit that will form the hemoglobin, right? And then
Yes,
you,
sir.
when you express the plant and grow the plant, this protein hemoglobin is expressed in the leaves and other part of the plant. So then the idea is that if you consume a fruit or leaf directly, now it's a very, like, it's a lab stage thing where you are, they are doing it in a tobacco plant and trying to figure out where the protein ends up, and it's mostly in the leaves that I know. So then one can think of a system where you can make therapeutic protein in plant and express them directly in plant and have it ready for consumption. Yes.
Okay, sir. So one more question,
Yeah.
sir, related to career perspective. Sir, do you support like direct PhD or doing masters then PhD?
Actually, I can take this question at the end of the lecture because half of the lecture is still left. So I will answer all these questions at the end,
Okay, sir. Okay.
not to deviate from the topic. Okay. Any more question?
Sir,
Sir.
my question is related to Alzheimer's. Can I ask it, sir?
Yeah, yeah. Go ahead.
Sir,
Yes.
like I want to make an aptamer for biomarker which can detect Alzheimer disease. So, sir, can you tell any biomarker which can in blood, which is present in blood, sir, for Alzheimer's early detection, sir?
Actually, my own understanding about Alzheimer is not that great because we work in Parkinson's, and my
Oh,
fellow colleagues work in Alzheimer. So my knowledge in that area is limited. But I can get back to you if you write to me separately, and I can ask my colleagues what they are working on. Yeah.
okay, sir. Thank you, sir. Sir, one more question, sir,
Yes.
about the receptor, sir.
Mm.
That means how receptors are inhibition and activation work. Like both have the same structure, but in inhibition, inhibitor inhibits because it has the same structure, sir, but it don't induce the conformational change. Or what is the, sir, means mechanism of inhibition and activation?
Yeah, because as far as I know, this receptor ligand interaction, or the enzyme inhibition theory works in the on the same principle. So it will essentially block the ligand site, and a receptor ligand, when they interact, the receptor ligand site will be blocked, and there will be no more chance for uh, triggering an immune response in this case. So that's how inhibitor differ uh, from from the elicitor, or the or the element that triggers this response. So it kind of doesn't allow the downstream signaling to happen properly. The downstream signaling is blocked, and then you don't see this effect.
So means it just occupy the site. It does not
Yes.
induce any conformational change.
Even if it induces conformational change, it doesn't do it in a way that it will allow downstream signaling. So a crooked conformational change, you might say, because anything, when it binds to any other thing, will eventually trigger some kind of a structural change, but they are not the same as the one triggered by a elicitor. Yes.
Okay, sir.
Good afternoon, sir.
Yes, Lisa. Go
Uh,
ahead.
yes, I have a doubt that as we said that protein has different stages, like from first to quaternary stages, and
Yes.
the more higher the stage, the more stable will be the protein. Am I right, sir?
Yes, like kind of organization, but when you talk about the protein, you are actually talking about, if it's a globular protein, you are either talking about tertiary structure or a quaternary structure. Those are
Okay,
oversimplification,
sir. So I
like
just wanted to.
yes. uh, yeah.
Yes, sorry, sir. Your voice got stuck. Sorry, sir. Can you just repeat
Yeah.
that?
Yeah, please go ahead, ask.
No, that's what, sir. Like now, my question is, why do we don't see such kind of stages in lipid and carbohydrates, sir?
No, see, like what I am essentially telling you is I am oversimplifying the process. It doesn't happen like that. Everything is like a cascade. So what I want to say is that protein are made of amino acids, and amino acids are like letters. Okay, you don't see it discretely, and then these letters form words, which is like in the form of alpha helix and beta sheet. But actually there is no literal translation of this all happens simultaneously. Just for your understanding, I am breaking it into four different stages. Yes, and you see this kind of arrangement with also sugars and uh, lipids. There are hierarchy, but it's not exactly the same as protein. Yes, because
So as we
yeah,
okay,
lipids and sugars, they do not form uh, they do not follow this scheme of like DNA, RNA to protein. So they have a different method of getting synthesized and assembled within the cell. Yes.
so when we can rupture a cell wall by using detergent or enzyme or
Yeah.
yeah, these two methods, then why we need such kind of expensive instruments like sonic by using sound waves and all.
Yeah, because sometimes you need to work with bulk. You are not working with 1 milligram or 1 ml or 2 ml of the cell. Supposedly, if you are working with 10 grams of cell or 20 grams of cell, then it will become a tedious task. Also, the volume, you will have to have a large amount of volume to work with. So I think the reason behind this is that, and also when you do it chemically, it's not 100% efficient. Some cells which will have accessibility will break first. Then you have to standardize a lot of things. It might work in a small scale. For large scale industrial setup, you will need something to break, because there then you need to purify protein in grams, not in like micro and milligrams. Yes, that's why you have this expensive setup. Yes,
Thank you, sir.
thank you, Risa. So if there are no more questions at this point of time, or I can take it at the end of the session, I move to the phenomena of column chromatography. So far what I have told you is that you you can express your gene in a heterologous host, like a bacteria or a yeast cell or a plant or animal cell, and then you have to get rid of the debris and collect the supernatant protein. And once you collect this supernatant protein, it's not only your protein, but there are lot of things that are present in the soup. And here, no matter what is the starting source, it's plant or it's bacteria or it's from eukaryotic cell, all has to go through the process of chromatography to separate the protein of your choice. So more or less, there are four different ways in which you can you can separate your protein, and they go in combination with one another. First is the size exclusion chromatography. The size exclusion chromatography is a very simple technique, where you, as the name suggests, that you exclude protein of certain sizes and allow protein of certain sizes to pass through the column. I'll go a little bit in detail in the next slide. Then you can separate the protein based on its physicochemical properties, especially the charge on the protein. How are the charge of the amino acid distributed over the surface of the protein? And this you do through ion exchange chromatography. And you can either have a cation exchange or anion exchange. Then you can purify protein based on the hydrophobicity of the protein. And essentially, this is not one technique that's used quite a lot, but we used it for some protein. You need to use this technique, whereby you bind the protein in a condition where the protein is completely unfolded, uh, and then you try to reassemble the protein by decreasing the salt concentration. And in the due process, the proteins with different hydrophobic indices will be separated separately. And then you have a biorecognition or ligand specificity, and this is called affinity chromatography. And one form of affinity chromatography is immobilized metal affinity chromatography, IMAC, which is kind of a backbone of most of the labs today who are working with recombinant proteins. Uh, so gel filtration or size exclusion, ion exchange, and affinity chromatography. These are the three basic chromatographic systems or processes that you need to understand when you tend to purify the protein or when you attempt for a protein purification. In size exclusion, what happens that the column, and now it's a oversimplification, I would say, because now most of the columns are pre-packed, they are commercially available, but you can do it on your own, that you buy the matrix, pack it onto a syringe or anything. Like in India, we used to pack it in the syringe or man-made columns, like we used to customize our own column. But you can buy those columns, and then you have beads of certain size which are sitting here. So what will happen that if you have a mixture of protein that you add into the system, there are certain protein which will enter into the bead and the pores uh, uh, of the matrix, whereas there are other proteins which are too big to enter into the pores, small pores in the beads, and they will come first. The place where all the proteins are excluded in the column is called big proteins are excluded is called the void volume. So anything bigger than that size of the protein will be excluded from the column, and then the small protein uh, which which kind of get entangled in these small pores will eventually come in the end, and you will have your protein of interest uh, getting resolved in the column after the void volume, and you can collect that protein and work with it. This is one method. Another method is that immobilized metal affinity chromatography or affinity chromatography. So here what you do is that when you are designing your construct or the gene of your interest, you create kind of a hook. And the hook you create is that it's a well established and studied system, is that you add certain like more than 6 to 10 residue of histidine either in the N terminal side of the protein or in the C terminal side of the protein. And when you express this protein, what will happen that this histidine will also be expressed along with the protein. So there is a kind of a histidine tag which is sitting with your protein of interest. So when you load it onto the column, what will happen that in the cell, there will be very few proteins which will have this stretch of histidine along with it. Maybe by chance they have 3 or 4 or 2 histidine sitting together, but not six stretch of 6 histidine. And this stretch of 6 histidine has been optimized, and it has been thought that it helps the protein to bind specifically to a matrix which is called nickel NTA. So it's a metric where you have nickel ion, and nickel ion uh, interacts with the histidine that's present in the protein, and they form a good complex. So using that technique whereby you break open the cells and you take the supernatant after removing the debris, and you add the mixture onto the column, and this mixture when it percolates through the column, what will happen is that the protein which doesn't have tag will come first, and the protein of interest will interact with the ligand, in this case here the nickel NTA, and it will sit in the column, and eventually you can break this interaction by either changing the pH, which is not recommended in most cases of the protein. So you have a molecular mimic of histidine which is called imidazole. So you give a gradient of imidazole wash, and then your protein is essentially chalked off from the column. And this is how, like if you automate the process, this is how the chromatogram looks in the system. So initially when you load the protein will have most of the protein have either tyrosine or tryptophan residue. So absorbance will go very high. This is the case where you are doing your sample loading. When you start washing, what will happen? That all other protein will get washed off. So you have a drastic drop in the 280 nanometer absorbance, which is due to the tryptophan, tyrosine, and phenylalanine present, mostly because of the tryptophan. If the protein has a tryptophan or several tryptophan, you will get a very good readout in the system. But there are protein which doesn't have a tryptophan, and for them you can monitor at a different wavelength. And one wavelength is like 214, where you are monitoring the peptide bonds between the protein, the uh, the absorbance of that. So then you start washing, and after the wash, what you do is that you have a 0 to 100% uh, gradient of the imidazole, which is kind of a molecule similar to histidine. So it will allow the histidine to get knocked off, and through competitive binding, the imidazole will bind to the column, and your protein will be removed out of the column. So once this protein is removed out, you will get, like in most of the cases inside the lab, and this I am telling you with the experience, that you will get 95% pure protein at this stage. And if you are working with a recombinant protein with a tag, this is the first method of purifying a protein. Fairly easy. You just break open the cell, load the supernatant into the column, you have your protein in your hand. Now, one bottleneck with this protein is that you still have this tag either sitting in N terminal or C terminal side of the protein, that you need to cut off, because you don't want like that 6 histidine to interfere with your structure function study or whatever you want to do with that protein, whatever downstream you want to do. So you have to remove that thing. So there are ways by which you can like you can have a protease site engineered between the 6 histidine and your protein of interest, which can be knocked off later on. Then you have ion exchange chromatography, and I am a great fan of this technique, because here, if you have a basic understanding of how your proteins are organized and what is the PI, the isoelectric point of the protein, then the this technique allows you in for a very quick purification of the protein. So uh, either you can have a matrix which is positively charged or a negatively charged matrix, and based on that, there is a there are different metrics, and I am not going to name them. There are some quaternary ammonium salt which forms the backbone of anion exchange, and there are some salt which have either act as a weak cation or strong cation, and that forms the basis of cation exchange chromatography. So you have these pre-packed columns that are there, and you just load your protein in a buffer condition, which is either, because if you load your protein in the same buffer condition as the pi of the protein, it will precipitate because of the, it's called isoelectric precipitation of the protein. So it's also a very good technique. When you want your protein to be precipitated out from the system, you just bring the protein to its pi and it will precipitate. So you don't want that. You want the protein in the solution. So you just move a pH ahead, and essentially most of my protein has a pi around 6.5. So I use anion exchange as a first step to purify the protein. So this is also a method of choice. And then at last is the hydrophobic interaction chromatography, which is a technique of choice when you want to purify protein which differ in their hydrophobic indexes. And as I told you before, is that it's also a kind of ion exchange chromatography. Here you do it in a reverse order. Initially, what we do in ion exchange chromatography is that we take two different solutions, buffer A and buffer B. In buffer A, we have a protein sitting in low salt concentration, and buffer B is just a buffer with high salt concentration. And then we give a gradient from low to high salt concentration, and the protein, uh, depending upon its charge density on the surface, it's eluted differently, and you have to run those fractions on a gel to see where you have your protein of interest. Alternatively, for hydrophobic interaction, it's based on which protein is more hydrophobic on the surface. What you do is that you incubate your protein in a very high salt concentration, and eventually you drop down the red line, you see the salt concentration slowly. And what will happen, based on the hydrophobic indices of the protein will be separated in different peaks. And here you have cytochrome C, RNase A, lysozyme, and trypsin as a representation. And this is how the protein will be separated. And then you can collect these peaks, check them, and then you have like from a mixture of protein you have supposedly, if you want the RNAs or if you want to work with lysozyme, you can just separate it through this process. So here what comes into play is the surface hydrophobicity, uh, which can be exploited to purify the protein. So with these three techniques in hand, the ion exchange chromatography, size exclusion chromatography, and mm, affinity chromatography, or you say immobilized metal affinity chromatography to be very precise, you can have or design a separation strategy. And separation strategy is actually three component. First is capture. So essentially the first part of the lecture where we were talking about how do you break open the cells, what kind of buffer you would like to take in the system to do this, those things are taken into account, the capture thing. Then you have an intermediate purification, and then after the intermediate purification, which is basically nothing but it's it's different strategy of column chromatography that you need to use. Sometimes you have to use ion exchange first, followed by a IMAC, sometime IMAC, and like affinity chromatography, followed by ion exchange. And the last part is polishing. And polishing is always the final step. And in this step, essentially what we use in lab is a final size exclusion chromatography to ensure that what we are working with is not a higher oligomer or something is sitting with it. We just want monomeric protein or a one population homogeneous protein to work with. Because if you work with a, say, a heterogeneous system, then the result would vary every time, and it will be uh, a great pain to analyze all those things. So essentially, like every single experiment that we plan uh, with a protein, this step is very very necessary. So when I purify the protein, I do capture, intermediate purification, and first step of polishing, and I store them. And when we start doing the experiment, we go for this once again. So if you see this scheme, this is like a trade-off between the speed, capacity, recovery, and resolution. So preparation, extraction, and clarification is the first step, and this is the basic scheme. What you do, you grow the cell, express the protein, lyse the cell, and remove the cell debris. This is the first step. Then you want to capture the SIP in the SIP method. So for the capture, what you do is that you can either use what I was calling uh, reversibly precipitate the protein using ammonium sulphate or some organic molecule. And here the idea is to isolate, concentrate, and stabilize. But sometimes you don't need that. You just move to intermediate purification, especially when you have a clear supernatant and you know that the protein is a soluble protein. You can just go for a IMAC, like immobilized metal affinity chromatography, if the protein has a tag. If the protein doesn't have a tag, you can choose one of the ion exchange chromatography and get fairly good level of intermediate purification at this step. And then you move ahead doing the polishing. Polishing means you pass it through the final size exclusion chromatography stage to have your protein of interest, which you can work with. So in the due process of all these steps, say you start with 30 to 35 milligram of protein here, at the end of polishing, you will be ending up having 1 or 2 milligram in hand of the purified protein. So that's why I call it a trade-off, because at each step of purification, you will be losing significant amount of the protein that you have to consider when you design. So you have to minimize the step when you go for that. So I already talked about this, that in how do we capture the protein and how do we do it is either through, depending upon the system, you use IMAC or you use ion exchange chromatography. This will be like you try to bind your protein to the column and remove the contaminant at the first stage of purification, after you have clarified your sample and have your supernatant in the hand. Second is the intermediate purification. Intermediate purification also involves certain steps of ion exchange or affinity chromatography. And after this step, you will have quite clean protein in your hand to work with. And then what you can do is that you can you can go for the next step of purification, which is the polishing of the protein. And what you aim for is something like this. You start with something like this. You start with a jungle, and then you go for a specific plant. If I just talk in a common parlance, so you want to remove everything that is there, just you want to have your protein of interest. And having said that, if you see this gel, there are still a small component lying here and there. So every time you have to undergo, like in the polishing step, you have to load this protein. It's just a representation. And then when you load this protein, these population of the protein will be dampened or removed from the system, and you can be sure that whatever you are working with is 95 to 99% pure. But it's just a myth. If you do a more sensitive staining of this gel, what will happen that you will still see some small amount of protein sitting there, but largely it's your protein in hand. And that's why therapeutic proteins are dealt with a lot of I think it has to undergo many steps before you get it as a therapeutic protein. And the idea here is to remove the oligomeric counterpart. In our case, basically we work with a protein called alpha synuclein, which is the main protein thought to be involved in Parkinson disease. And you see those protein in the plaques from the patient. So this protein is a random coil or intrinsically disordered protein. To purify this protein, if you purify this protein, what will happen? As soon as you purify, this protein will form certain oligomers. So to remove those oligomers and have a homogeneous population, you need to do a size X. So how do you check the protein? Finally, when you have the protein in hand, and after each step you have to do this process, you do it's called a denaturing page or SDS gel electrophoresis, whereby you load your protein samples and you run it uh, under the influence of electricity, and then you get the nice separation, and you are aiming to get your protein of interest purified as a single band. This is your aim. So then I would like to thank the organizers, and I would like to talk about two companies here. And interestingly, Mycorrhiza is a company which is a startup by a PhD student here, who has not genetically engineered any strain, but he's using some super strains of algae, which can be used as a food additives. And they have a pilot project running, and it's a startup which has got a significant amount of grant from university and many of the startup projects in Europe. And this is run by an Indian actually, and they are trying to have a alternative to like good protein contents for vegetarian and vegan people in the form of algal supplement. Even in India, you have company like Murgappa Chetiyar that make spirulina, which is a feed product or which can be consumed. And then there is another offshoot. These things I am telling you because to give you an insight that where can protein engineering or protein purification or protein as such can lead you, apart from basic research and understanding disease. So there is a company called Biopetrolia, and they try to make they are making many products. Actually, they are making essential oils, they are making um, alternative to fossil fuels, and they do so by engineering the yeast cell. And this project is also kind of it's a university incubated center, and they are working to find alternative to fossil fuel and green energy and other stuff. So at the last, I would like to thank my professor with whom I am working. She's Pranila Vitung staff shade, and we work in a Chem Bio division, which is part of Department of Biology and Biological Engineering at Chalmers University, Sweden. And then I would also like to thank Professor Gupta, who has been a constant source of encouragement for me since my college days, and I have been following his great work in grassroots innovation, and have been in constant touch. And he has been very instrumental in shaping who am I today. So thank you, and I am open for questions now, if there is time.
Hello, sir.
Yes, Divyansh.
Sir, I had a protein sequence and I used eye tracer to predict its structure.
Okay.
So, sir, my question is that how much I can confirm that it is the same structure as the real structure?
So, as I understand that you have a protein for which the structure is not known, right?
Yes, sir.
And then you do a prediction based on the. So these prediction models are based on like the structure which are similar or which are already deposited in PDB, right? So you cannot be like 100% sure, but like that's why there is you need to bridge the bioinformatics side to wet lab side to see, okay, say if you are designing some inhibitor or doing some stuff, is that really relevant in the real context when you want to use it?
Yes, sir. That's what I'm asking, sir. How?
Yeah, so well, like then you have to go for a proper kinetic study of receptor ligand interaction, and it's a part of bioinformatics. I don't want to go into detail, but you have this QSAR, and you have this docking and molecular modeling stuff involved that you need to follow to get an answer from the bioinformatics side. And once you have the bioinformatics side done, you will eventually have to move into the lab and see whether the things like the binding energy or the docking environment that you create, is it really relevant in the real situation or not?
Yes, sir. Thank you, sir.
Thank you. Yeah, there was some career related question. Uh, if you can repeat once again, I can just take that question now.
Sir,
Okay, Nikhil, continue.
Yes, Nikhil.
sir, can we use gel filtration chromatography to find out the molecular size of the, like molecular weight of the protein or
Exactly.
enzyme?
It's very easy to do. So the way to do it is that you can, if you have a column, you can run certain proteins of which you know the molecular weight before. Like they are called protein standards. You just run the standard, and after running the standard, you run your protein and see where it falls. And by simple calculation, you can compute the molecular weight of unknown protein. It's a standard technique uh, to find out the molecular weight of any unknown protein, provided the column has the capacity to resolve your protein. Yes.
Sir, how is the standard run, sir? Like based on the time at which it elutes out faster or slower, based on that?
No, so actually standard is nothing but it's a mixture of protein. So you have a protein which has a which are of different molecular weight. Say if you want to look for an unknown protein which is 30 kilodalton, say you run a standard which has a mixture of 10 kilodalton to say uh, 80 kilodalton, and also a protein to a protein or bigger molecule which is far much bigger, to find out the wide volume of the column. So once you know the wide volume of the column and you run standard this standard will, if you have a mixture of four protein, these four protein will elute separately if they are globular, and based on that four protein eluting separately, where does your protein elute? So you can either compute as a function of the we call it v by v0 value, and uh, then you have the log of molecular weight. And when you do it, it's a very simple calculation, so you can calculate how where is your protein coming in
Sir,
simple excel. Yeah.
sir, actually, Stokes radii or some parameter is used to find out uh, the molecular weight of the proteins that are diluted from gel filtration, no, sir? Stokes radii.
Yeah, but you don't need to go in those over complication or calculation. You just need to follow a simple thing, that you just like the way you do it for what you will do is that you when you when you compute this standard with four or five different components in it, you will get a linear graph, and in that linear graph you just need to extrapolate your point and you get your molecular weight. That's the way we do it in the lab. But yes, there are ways to
Sir, for
yeah.
sir, for example, if standard is run and that resolved into some like if you have 30 kilodaltons protein, that is the
Mm.
largest molecular weight in the standard, so
Yes,
that elutes out faster than the smaller ones, right, no, sir?
that will the bigger protein, uh, the biggest protein will come first, which will be the void volume. And what will
Yes, sir,
happen in the
because
size exclusion is that first you will have the bigger protein coming, and then you have the smaller protein coming. Yes.
sir, it can so happen that the smaller proteins also enter the interstitial space, and one that elutes out faster may not be the largest protein, no, sir?
Can you come once again? What did you say?
So like it can so happen that the smaller proteins also enter the interstitial spaces and come out faster than the larger proteins, no, sir?
It's least likely, because if you are using the right kind of column, what will happen? If the protein is small in size, if it's globular, it should ideally enter the pores of the matrix. Otherwise all the protein will come at void volume, because it's beyond the resolution capacity of the column. It will
So
not
like
be
how is
here and there.
this assurance made, sir? Like how it is assured that the smaller particles do not come out faster?
It cannot, because the way to check it is to run a SDS page gel and see whether the protein that you are aiming for, where it is eluted. And the other thing you are not considering is that maybe the protein is small, but is the protein aggregated or not? If the protein is aggregated, say this smaller protein in theory can be 20 or 30 kilodalton, but in practice, on column, what is happening is that this protein is a very big protein. It's a oligomer where many monomers are associated together. That's why even if you get your protein like a bigger protein, it's it's a assembly of many molecules. It's not monomeric.
Yes, sir.
This can
Like,
happen.
sir, if
Yeah.
you're using gel electrophoresis, sir, where the molecular sieving of the agarose gel is used to separate DNA components, no, sir?
Yeah, but that's different. And you here
Yes,
you
sir.
use
Like here also molecular sieving is not used, no, sir. The principle is completely different, I think so.
the size exclusion and the SDS page. I'm not telling agarose. I'm telling polyacrylamide gel electrophoresis.
Yes, yes, sir. SDS
So
page.
yeah, so in both the cases, the principle is essentially the same.
Sir, if principle is essentially the same, then
Yes.
in SDS page, larger molecules are retarded, whereas the smaller particles move faster than the larger particles, no, sir. Whereas in
Yeah,
gel
because
exclusion,
there you mask all the charges, right? And also you break the protein. Like if there are oligomers, what will happen? Due to the SDS, you will break this interaction. So you only see the monomer. But on size exclusion chromatography, you will also see monomer and oligomers of the same protein.
okay, yes, sir. Understood. Thank you.
Yeah. Any more question?
Yes, sir.
Yeah.
Uh, s- Sir, as you mentioned in the hydrophobicity interaction chromatography,
Yes.
sir, how is temperature dependent on that chromatography technique, sir?
Usually, I cannot comment much on the temperature dependence, but when we do these processes, usually we consider that most of the protein would like to be in the ambient temperature, or we carry out the purification procedure in cold. So I don't have the idea how what exactly you mean about temperature in terms of hydrophobicity. Yes. Is there any connection? If there is, then you can tell me. Uh, I really don't know. And that's the least used technique, because most of the protein you can purify with the three processes that I told you.
Okay, sir. Thank you.
Thank you. So if there are no more question, or if you have any question, you can write to me directly. You can get my email ID from the organizers, and then you can write me directly. If there are career related question, I will be happy to answer. There is no issue. uh, I like in general, I can tell you that the PhD in Europe is very rewarding. Although I have not gone through a PhD here. Yes.
Okay, sir. Ambika, you want to ask anything?
Yes,
Mm.
sir, I asked you in initials that, so what do you think is better, sir, doing masters than PhD or direct PhD options are also there. So what should be preferred?
Like it depends. If you have a 4 year bachelor's degree, I don't know which way you go. So
Yes,
if
sir,
you
I
have a
have a 4 years bachelor's degree.
if you have a 4 year bachelor degree, I think the process is quite simple. Actually, you can come for a 1 year master's degree abroad, and then if you have the right kind of research acumen, you can be recruited directly for a PhD position. So
Okay, sir.
initially you will have to go for certain training, because usually for a PhD recruitment, what happens is that uh, you need to build certain kind of trust, right, in the lab, and that requires some time. So if you go for a 1 year masters where you do, say, here you have a flexibility to start directly, you do 1 or 2 credit programs of say 15 credits or so, and then you can move directly into the lab and do lab work. That flexibility is available here, and that will give you an opportunity to interact with the scientist and then eventually get recruited.
Okay, sir. Thank you, sir.
So if
Yes.
there are no, yeah.
Subrajit, you want to ask your
Yeah, hello, sir.
Yes, Subhrojit.
Sir,
Go ahead.
so actually, I have a question, a research-oriented question, sir.
Yeah,
So
please.
actually, sir, there are metalloproteins in our
Yeah.
body. Okay, sir.
Yes,
So
yes.
have you come across any papers or any research, those who target using non-invasive IR, infrared to
Yeah.
detect the metalloproteins of body?
uh, that is very specific question, and I cannot answer this, sorry, Subhrojit.
Okay, sir.
This area is a little bit means I don't know. We work with metalloproteins definitely, because we work with copper and copper interactions with protein, but the technique that we use is not IR. We use certain other. We use circular dichorism. We use EPR. We use some other techniques. Yes.
Okay, sir, but not IR, not yet.
Not IR. Not IR. I don't like in our group we don't use it. But
Okay,
we
sir.
we definitely work with copper in cancer and metalloproteins, and how does the this homeostasis of metals lead to disease malfunction. And both of the disease that we study in our lab, Parkinson as well as Wilson's disease,
Okay,
uh,
sir.
which was earlier studied in the lab. So these two diseases have a direct implication of metals, because the proteins
Okay,
that
sir.
are involved can bind with metals. Yes. Yeah.
Yes, so actually, circular dichromism, I have also seen the approach of circular using circular dichromism. But
Yeah.
we have many types of IR sensors are available, means which can detect metals from means from far range, and they have high affinity to detect it. So,
Yeah.
so in metalloprotein, as we know, there is a metal binding with the structure. So
Yeah. Yeah.
means I was trying to write a paper like this in my second year, sir. So
Okay.
that's why
Okay.
I want to confirm it with it will be can I forward with this, or it's not possible? Or you
No, you can forward, but I don't have the expertise in that area. That's the thing. I am a hardcore biologist, so I will not be able to help you that much.
okay,
But
sir. Okay, sir.
but the idea is that for our metal detection process, or if you want to know the metal content of certain protein, we use a kind of mass spectroscopy which is called IPMS, which is
Yes, sir.
also very common
Yes, sir.
here. Yeah,
Yeah,
we
yes, sir.
are using that.
Okay, sir. Thank you, sir.
Thank you.
Okay, guys, so I'll share your email ID of Dr.
Yes.
Ranjeet if you have any query or if you want to discuss about your project. So thank you so much, sir. Again,
Thank you.
your session was really interactive and informative.
Thank
Thanks a lot.
Thank you, and have a good day ahead.
Same to
Bye.
you.
Bye.
Yes. Thank you, sir.